Okay, guys, so let's begin. We are responsible for knowing anatomy, anatomy, and physiology, and lab. So, anatomy, we mentioned, anatomy is the study of the structure, and physiology is the study of the function. And in the lab, we're going to be studying the experiments between an independent variable and a versus a dependent variable, and more about that later. So independent variable always on the x-axis. This is the x-axis. We're gonna have independent variable, and then on the y-axis we're gonna have our dependent variable, and more about that later. Okay, so the structure and the function. There is a connection between the structure and the function. We mentioned that the knife is able to cut because it is sharp, and the hammer is able to hammer things down because it is dull. Even though both are made up of steel, but their function is different because their structure is different. Now, when I examine that I have a neuron and I have also an islet of Langerhans, that is the cell of the pancreas. Then I have a skeletal muscle cell, or a skeletal muscle cell. Or I can have a cardiac myocyte, or a cardiac cell, cardiac myocyte. These cells, always when you want to understand the difference between two things, always begin with the similarities. Begin with the similarities, then the similarities will help you find the differences. So, what is the similarity between a cardiac cell, a skeletal muscle cell, an islet of Langerhans, a neuron, a hepatocyte, a cell of the liver? Hepatocyte, the liver that looks like a bow. You know the bow and arrow, the liver, the hepatocyte. What is, if before I understand the difference, I must begin with the similarities. So what is similar between all of these different cells? They all have the same DNA. So if you go in the nucleus of the cardiac muscle cell, the nucleus of the skeletal muscle cell, the nucleus of the islet of Langerhans, the nucleus of the neuron, all of them have the exact DNA. Then why do they look different? Now this DNA, and this is very important, if you look into the cell, let's say this is the nucleus, and the nucleus is inside the cell. The nucleus has DNA, and the DNA is structured as chromosome, like this. This chromosome is made up of really long strands. I'm just untying it. All of this chromosome is identical in all of them. Then what makes each cell? Cell 1, the cardiac muscle cell, the skeletal muscle cell, the smooth muscle cell, for example. So many different types of cells. Smooth muscle cell, like the blood vessels, the blood vessel cells, the islet of Langerhans, the pancreas cells, the neuron cells, the hepatocyte cells. They all have the same DNA. Then why do they look different? Because just because you have the same DNA, it does not mean all of it is active at all of these cells. So the DNA from this region to this region, if you transcribe it, you will get a smooth muscle cell. If you transcribe a region, you will get a smooth muscle cell. If you transcribe this region, for example, you will get a cardiac muscle cell. If you transcribe this region, number 3, you will get a skeletal muscle cell. If you transcribe this region, number 4, you will get a pancreas islet of Langerhans. If you transcribe this region, you will get 4, the neuron. If you transcribe this region, you will get the hepatocyte, and so on. So that shows you, just because the DNA is there, it does not mean all of it is active. Only the part that gives the cell its identity is active. So what is the name of that part? What is the name of the part that upon transcription will give you the proteins that are needed by the cell? So remember we said that the cardiac muscle cell needs a lot of skeletal, cytoskeletal proteins, needs a lot of dynein, actin, myosin, all of these mechanical proteins to help it do its function of pumping blood. So that means if you transcribe this region, you will get all the proteins necessary for this cell. So this region that when transcribed will give us this cell is known as the euchromatin. This is the active part of the DNA that gives the cell its identity. The inactive part, so the euchromatin is the active part. The inactive part is known as the heterochromatin. So, in a cardiac muscle cell, the region of the DNA that gives it its identity is the euchromatin. All the DNA of the other cells is now heterochromatin. In the next cell, in the isolate of Langerhans, the region of the DNA that gives it its identity is the euchromatin, and the rest is heterochromatin. So what I'm trying to say is, just because you are euchromatin in one cell does not mean you will also be euchromatin in the other cell. And just because you could be heterochromatin in one cell because it doesn't need you, but you will be euchromatin. That heterochromatin can later on become euchromatin in the other cell. This is so important. Is this concept clear? Is this concept of what is the difference between euchromatin and heterochromatin clear? So now you must understand the central dogma of biology. The central dogma of biology. So what is the central dogma of biology says? It says that from DNA we get mRNA, and from mRNA we get our protein. What is the name of the process from which we get mRNA out of the DNA? That is known as transcription. Very good. That is known as transcription. And from mRNA into protein, that is known as translation. Excellent. Now, this is supposedly the central dogma of biology in this direction. Now go and tell that to HIV. HIV has a very dangerous enzyme that can go the other way around. It can create from mRNA DNA, and this very important enzyme is known as reverse transcriptase. So the virus does not have a DNA of its own. So when it hijacks the cell, it will go inside the cell and activate this very dangerous, scary enzyme, and it can now take the mRNA and reverse it against the central dogma of biology to give us a DNA. And that is how it's able to survive and cause your own cell to transcribe its DNA. So first it's going to go inside the cell, destroy the DNA of the cell, then it's going to activate reverse transcriptase to convert its own viral mRNA into viral DNA, then hijack your cell to go from to do the central dogma of biology, to go from viral DNA into viral mRNA into viral proteins that will destroy the cell. So you see how malignant and evil that virus is. It doesn't have a DNA, but it hijacked the system by activating from off into on its own dangerous enzyme that will convert the mRNA of the virus. So we begin at step one. Convert the mRNA of the virus into two DNA, and then go down that route to create viral DNA, viral mRNA, and viral protein. So here, this is just to help you appreciate that this is the central dogma of biology. Whenever we need a protein, and we have two types of proteins, because we have anatomy and physiology, we have proteins that are structural, we have structural proteins, and we have functional proteins. So some proteins are there for structural jobs, like your nails, the collagen. The collagen is there for structural integrity. The same way you have a fence around your house is for protection. But are all the proteins like collagen in your nails, is it only for structural support? No. There are proteins that have functions, like hemoglobin. Hemoglobin function is to shuttle oxygen from your lung that is highly saturated. So your lungs have very high partial pressure of oxygen. Well, that makes a lot of sense because you breathe. So air comes in, the lung has really high partial pressure of oxygen. Who has a really low partial pressure of oxygen? COPD. Very good. So patients who have COPD, they have a very low oxygen saturation even when they inhale. But that's not what I mean. I mean in natural. So COPD is when there is pathology, when there is a disease. So now we're not talking about disease. We're talking about in the natural state of the body. Naturally, your lung has a lot of oxygen. Who in your body has a little bit of oxygen? When you're running, your skeletal muscles. So you're running the marathon, your tissues, they need oxygen. Your tissues need oxygen, they need nutrients, and they need, so what's the function of blood? Blood has three functions, two pluses and one minus. The first plus is to give plus oxygen. So this is tissue. If blood comes to this tissue, it's gonna give it the first plus oxygen. The second plus is gonna give it nutrients. It's gonna give it food. We have three different types of nutrients. We have fats, known as lipids or cholesterol, and that once you give it to the tissue, the cell is going to be able to break down this fat into ATP. That's what we need in the end. ATP is the energy currency of the cell. So we're going to be studying in metabolism how we end up with ATP, from fats. So this is a holistic cooperative job, beginning with your blood. So your blood delivers when you have a meal, when you eat the steak. So the steak has a lot of cholesterol, fats, and lipids. It delivers it to the tissue, then the tissue delivers to the cells this broken down fats and lipids, and then the cell is gonna get ATP out of these fats. There's a biochemical pathway known as, you will learn that later on, but it's not too bad to just mention it now, known as beta oxidation. So we get fats, shuttle them in beta oxidation, we shuttle them in the beta oxidation pathway, and that gives us ATP. What if I don't eat fats? What if I eat carbs? What if I eat carbohydrate? Carbs, such as sugar. We have different types of carbs. We have the sugar of milk, lactose. We have the sugar of the oatmeal, known as maltose. We have the table sugar, sucrose. Well, no matter what sugar we have, we're gonna shuttle it into the cell, and obviously the system that gives us ATP from fats, can we use it also to get ATP from carbs? Can we use it also? So this is a system that gives us ATP from fats. Now we're not eating fats anymore. So that's one. We're eating two, carbs. Do I need the same system to give me ATP, or do I need my own unique alternative system to give me ATP? Because now I'm not eating the same thing. I'm eating carbs. So I'm going to need my own separate system because I'm eating a different thing. So I need a different system, and that is known as glycolysis. So when I eat my carbs, the cell will shuttle it into the system known as glycolysis in the end to get ATP. So we get our ATP, but with different systems. So do we only eat fats and lipids? Do we only eat carbs? No, we eat proteins also. Proteins. And proteins, when we eat them, we break them down, we catabolize them. Do I need my own separate third system, or do I use one of those all the ones from before? I did my I need a third system. Excellent. And that is known as the protein catabolism. And then I get ATP. So you appreciate that I ended up with my ATP from my different nutrients. So when you have a meal eating pasta, pasta is carbs. Pasta with meatballs, that's fats. Pasta, meatballs, and then you eat some form of protein. Use some form of protein, and then you mix them up together, and then you eat them. But once it goes inside your body is going to digest them, send them to your tissue, and the tissue is going to put each nutrient in an appropriate system to give me my ATP in the end. So I don't have a one universal system for all of them. So I break them down. That's what my digestive system does best, and that is to separate my nutrients, break them down from macronutrients to micronutrients, then shuttle them in their respective systems. How did that happen? That happened. I needed protein to do that. I need proteins to do that. This cannot be done on its own. But how can I get protein? I need mRNA. How can I get mRNA? I need DNA. So now you appreciate how DNA is responsible for making the proteins that sustain life. And we're going to be learning about what do these proteins do? They are enzymes. So all of these functional proteins, they are enzymes. They have a function. They are not structured, so they are structural proteins. We don't discriminate against our proteins who are not functional. We don't say, oh, only the functional proteins are the best. No, structural proteins are also important. There are many diseases where there is connective tissue problems due to the loss of the ability to, in the genesis and the making of collagen. So we want to appreciate both. But you know that hemoglobin, the same way the digestive system is responsible for providing our tissue with nutrient, the respiratory system is responsible for providing our tissue with oxygen. So that's what led me to get into this. My blood is providing two pluses and one minus. The first plus is providing nutrients. We talked about that. Providing my different types of nutrients, my fats, my carbs, my proteins to my tissue. That's my tissue. That's my hungry tissue that needs ATP. So it needs ATP from the nutrients. So the blood is coming to provide nutrients, and it's also providing what else? Oxygen. So that's where the hemoglobin comes in. So hemoglobin takes oxygen from an area of a high partial pressure of oxygen to an area of a low. Do you think the tissue has high or low partial pressure of oxygen? It has very low because it's doing oxidative phosphorylation. Oxidative phosphorylation. So you work, you get paid, you come home, you bring the stuff into your house. So you have a lot of money, high partial pressure of oxygen. Then you bring it home to the children, you buy the food, the cereal, or the stuff. That's what it's being consumed. So you take it from where there's a lot of it, and you deposit it where it's needed. So we take oxygen from the lung, where there's high partial pressure of oxygen in the lung, and there is really low partial pressure of oxygen in the tissue because it's being used there. That's how I'm able to walk. So I'm walking because my tissue is utilizing oxidative phosphorylation. It's eating up all that oxygen to enable me to walk. But after it does that, oxygen is gone. When you go spend money in the store, it's gone. You need more money. So that's who supplies the oxygen. Your hemoglobin supplies oxygen from the lungs to your tissue. Then the tissue will always have low partial pressure of oxygen. That's the waterfall. You know the waterfall, the water falls from the top to the bottom in Niagara Falls. It falls like that. So oxygen falls from the lung into the tissue where there is low partial pressure of oxygen. But that's not the only job of hemoglobin. Hemoglobin is a shuttle. It takes, is uptown, downtown. The subway doesn't only go one direction. So hemoglobin will not only go from the lung to the tissue. It has a cycle back from the tissue back to the lung. So here, what is it doing now? Carbon dioxide. So it begins with carrying oxygen downtown to the tissue. Then it has an uptown trip by taking carbon dioxide. So now we have another variable, carbon dioxide. Please pay attention. Where do you think the partial pressure of carbon dioxide the highest? Do you think there is a very high carbon dioxide in the lung, or there is a very high carbon dioxide in the tissue? In the tissue, excellent. That's where the minus comes in. So my blood gives two pluses. It gives my tissue oxygen to do oxidative phosphorylation to survive, and it gives it nutrients for ATP, 2 plus. And it takes away metabolic waste. Metabolic minus toxic waste, poison, carbon dioxide. So when I'm using all of this oxygen, I'm generating very high partial pressure of toxic, poisonous carbon dioxide. Do you think your lung has high partial pressure of carbon dioxide? Of carbon dioxide? No, because it just gets it out. So it always high partial pressure of oxygen and low partial pressure of carbon dioxide. So in our downtown trip, we take oxygen from an area of high partial pressure of oxygen to an area of a low partial pressure of oxygen. Do you see the downtown trip? From here to here. We take oxygen from an area of a high partial pressure of oxygen to an area of a low partial pressure of oxygen. Then, when we give the tissue oxygen, the seat is empty. The seat was occupied before by oxygen. But we already unloaded oxygen to the tissue. Now hemoglobin seat is empty. The subway is empty. Now we need to take back uptown, we take carbon dioxide. So we take carbon dioxide from an area of a very high carbon dioxide concentration to a area with very low carbon dioxide partial pressure so you can exhale it out. This is so important. Is that clear? Okay, so now we see that in our central dogma of biology, if I have a mutation in the region of the DNA that when I transcribe it, I'm supposed to get an mRNA for hemoglobin. When I transcribe it, hemoglobin is not working, like anemia, like sickle cell anemia. Do you think I'm going to be able to have my hemoglobin? So you see, the problem is here, is the central dogma of biology. I need to ensure that the enzyme, that the protein that I'm going to get, let's say I want collagen. Rest assured, if I'm going to get collagen, there was a DNA region for collagen, DNA collagen, mRNA collagen, protein collagen. If I need hemoglobin, there is a region in the DNA for hemoglobin. Then there is a region in the mRNA for hemoglobin. Then there is a region in the proteins for hemoglobin, and then I'm going to get my hemoglobin. So all of the pharmacology now they have found out that this is called a deconstructionist. We're gonna see it in the slides later. There is a deconstructionist school of thought in our anatomy and physiology, that if we want to find the cure for cancer, we want to find the cure for disease, we have to go from the bottom up. We have to go all the way to the cell. They have learned that the cell is the unit of life. The cell is the unit of health, and the cell is the unit of disease. So I am healthy because my cells are healthy, and I am sick because my cells are sick. I am alive because my cells are alive, and I am dead because my cells are dead. So now they used to look at things holistically. They used to look at things from the outside, say, well, examine the signs and symptoms, but they realized that this signs and symptoms was all because that small little part mutation in the DNA. Little part mutation in DNA. So now they're talking about how to repair that, how to repair the DNA, how to make sure that you don't even get a mutation to begin with. Stay away from the ultraviolet radiation. Stay away from the carcinogen. Stay away from asbestos. Stay away from chemicals and physical agents and chemical agents on some infectious diseases like the some virus lead to cancer. Yeah, like the pap smear. Females have to go and do pap smears. In the pap smear, you have the human papilloma virus, HPV, and that's a virus that lead to cancer. I mean, it's unbelievable. They used to think that there's three different types of disease. If you have a disease, it's one of three. It's either a congenital disease, something you're born with, you're born with genetically, something wrong, you had no control over it. It's embryological development malfunction. There is an extra hole in the heart. There is a disability in the fusion of some parts of your embryological tissues. So they said that the disease can either be congenital or inflammatory. So inflammatory is like when you have a trauma or a laceration or an infection, and you have five cardinal signs like heat, redness, pain, swelling, loss of function. So whenever you have these signs, whenever there's redness or pain or heat or swelling, that's the inflammation. So we began with congenital types of diseases, two inflammatory types, and three is what? You tell me this one, you're really smart. What's the third type of disease? So if you're sick, if you have a disease, it's one of three. It's either a disease that is congenital, you are born with it, a structural defect that you're born with, a structural mutation you're born with, or it could be an inflammation. You got some infection or some trauma or some laceration where there's a lot of redness, heat, pain. In congenital disease, there is no redness, heat, pain. In inflammation, there is redness, heat, pain, and swelling and loss of function. What do you think is the third one? So autoimmune, very good, but autoimmune is a type of inflammation, because what happens in autoimmune? You have your immune system has amnesia. So your immune system doesn't know enemy from friend. I'm sorry? Okay, I'm gonna get to that. So contraction, hold on. So let's deal with immune system first, then we're gonna talk about contraction of diseases. So immune system, what happens? It's when your own body, in some countries the military kill his own people. So that's the same thing what's happening in your body when your body has these antibodies against the cartilage of your own bones, of your own knees. Exactly, like rheumatoid arthritis, or like diabetes type 1, where a baby is born and there's some mutation where the antibodies are blind and they destroy the DNA. I mean, your own immune system destroying the DNA. Like lupus also, excellent. But that's a type of inflammation because later on, so pay attention, what happened with rheumatoid arthritis? Is there pain? So that's one of the five. Pain, heat, is there heat? Is there redness? Is there swelling? So that's inflammation. So same thing with contraction of disease. So when I contract the flu, when I contract a viral infection, a parasitic infection, a bacterial form of infection, let's say I contracted a worm infection, does that infection lead to my cells being damaged, my cells red, my cells hot, pain? Yes. So when you have the flu, don't you feel like some pain in your? Yes. So there's pain, there's inflammation. So that's a type of inflammatory disease, and that's a type of inflammatory disease. What's the third one? So structural or functional can become. So I can show you a congenital disease where there is a structural defect. So you are born with a neural tube defect. You, I'm sorry? Yeah, cleft clip, yeah. So that's a structural defect that led to congenital disease. Rheumatoid arthritis also can be a form of structural defect because my antibodies are lacking the region that helps them identify those antigen presenting cells, self from non-self. So being structural or functional is not a type of a disease. They lead to one of the three types. What's the third type? I mentioned it many times already. I mentioned it. So if you have a disease, it's one of three. Congenital, you're born with it. Inflammatory, it's a form of inflammation. What's the third one that you can get? Yeah, so mutation leads to what? Cancer. Excellent. Excellent. So tumor. Tumor. It could either be, so three, it's a tumor that is either benign or malignant. Now, these are the three types of categories. In the beginning, when they develop, they can cross. So, for example, you have a tumor. This tumor can progress, invade, destroy nearby tissue, and it turns now from a tumor disease into inflammatory disease. You know, cancer patients have part of their tissue damaged. So it began as cancer, painless. So that's why cancer has no pain, because it begins as non-inflammatory. So cancer is a painless swelling. But then when it grows and it metastasizes and it invades nearby tissue and travels in the blood to far side and then stick to other far regions in the lung and then it invade and destroy the lung, then it becomes inflammatory. Because now the cancer patient feel pain now. But that's the later stage, that's the last stage, because now we have moved from being cancer into being inflammatory disease. Excellent. Can you think of the opposite? Can you think of an inflammatory disease that becomes cancer? I mentioned it already. It, yeah, excellent. So HPV, you guys said that, human papillomavirus. Yeah, that begins as a viral infection. And we mentioned already that infectious disease lead to inflammation. So it begins with the infection, with the pain and all of that. Then the pain goes away, and then there is painless cancer. So they begin like that, but then they cross. And you will see these relationships as we go in the system. Um, what is there? Okay, so are we through with structure and function and how the structure is dependent on the function and the function is derived from the structure and how we use the DNA to the difference between euchromatin and heterochromatin, all that stuff. We got that part. Yes. Tumor. So the tumor, there's two types of tumor: benign tumor and cancerous tumor. So these are the three types of diseases that we begin with. But if we don't hold it in time, it progresses to very bad. Is there an eraser here? Okay. There's wipes. Are they supposed to cut somewhere? Okay. Oops. Okay. All right. So now we're gonna talk about how we're going to talk about the destructionist school of thought. Remember, I mentioned that. I mentioned there is a deconstructionist. We break down the structure from an organ system into tissues, into cells. Then we go deep inside the cell to see all the organelles. Okay, so um, is it the time for the student break? Never. It's just a little. Okay. It's I've been talking for an hour. Wow. Yes. Okay, go ahead. Go ahead. No problem. Okay, this is very important now. So, the concept of stress. Yeah, you have to you can find it on the you can find it on the recording. Remember, you see what I have here. This is not the Family Feud. This is the this is the recording. Okay, okay. So go find out because before they tell you you're not gonna get credit for being here, and then you got to be somewhere else. Yeah, I've seen you, but maybe hold on. Maybe they're gonna tell you you're not supposed to be in this class, and then you're gonna go to you're supposed to be in another class. So go to them. Go find out from them. Yeah, yeah. Go, and then you can find this in the video. Yeah. Okay, I'll hold it. Okay. You can watch it later. Okay. All right. So the concept of stress is the spark. So fire begins big. That's where the deconstructionist theory comes in. That we begin with small things that lead to big things. So the fire in the wood, it began with a little somebody throwing a little cigarette, or maybe a little small spark of fire that led to a really big fire. Same thing with the disease. So in the disease, you don't begin with chronic you don't begin with chronic inflammation. And you don't even begin with acute. That's the step before chronic, yeah. Acute inflammation. Well, you see, it's a long line. So we begin with the stress. Give me an example of what a stress is. Stress could be a physical agent. Stress could be a physical agent. A physical agent, what's an example of a physical agent? Nursing school is not a physical agent. That's an emotional. Actually, that's it's called moderate stress. We do need some moderate stress. Exercise. When you run on the treadmill, that's an example of a very beneficial moderate stress. So today we're gonna correct our concept of what stress is. Some people think stress is something that you never have to deal with. No. Today we're gonna learn that positive moderate stress is actually important for you. And people that do not do moderate stress, they have a lot of problems. So, for example, they brought two models of rats. So, a model is what we do our experiment on. There is a mouse model, there is a cat model. So, in the mouse model, what they did was they suspended a rat, and then they forced another rat to run on that circle. And then they took the x-ray of the bones to measure the bone mass of the two types of mouse models. The problem was, after a week, they realized and they had a control. Obviously we need a control. So a control is a group that we do not test, we just give them a placebo. Then there is a group that we are doing our independent versus dependent variable. So we have an independent versus dependent variable. The test the effects of exercise stress on bone mass, right? So we have three groups. One group, they do not, we just leave them rock, do whatever they want. Then group two, we suspend them. So we devoid them of exercise stress. There is no exercise stress. Three, we add to them exercise stress. And then we measure with x-ray to see the bone mass. So here, who's the independent variable and who is the dependent variable? One is independent. No, one is the control. That is the group that we do not stop them from exercise and we do not make them exercise. We leave them to their devices. They run around. Yes. Very good. So the exercise, so we have two things: exercise A and B, bone mass. So obviously, one of those two is the independent variable. It's a variable. Why do we call it variable? It's a variable because it can go up or down. Good, change. Meaning it can increase the independent variable on the x-axis and the y-axis. So when we have something like this, something like that, that represents for us that the independent variable always on the x-axis can increase and decrease. That's why we call it independent variable. We don't call it independent constant because it's not constant. It's changing. And we call it dependent variable because it can change. It can go up, increase, can go down, decrease. So the exercise. So the law says independent variables are not what I am trying to measure. The dependent variable is what I'm trying to measure because it is dependent on the independent variable. My bone mass is my dependent variable because my bone mass is dependent on whether I exercise or not. So my bone mass is going to go on the y-axis. So that's my bone mass. It's my bone density, bone mass. And on the x-axis, I'm going to have my independent variable, and that will be the exercise. Do I exercise a little bit? Do I do 10 cycles in a minute, or 20 cycles in a minute, or 30 cycles in a minute, or 40 cycles in a minute, or 0 cycles in a minute? That exercise stress is my independent variable, but my bone mass is dependent on it. So they realized that the group that had a moderate positive stress was able to have very strong bone density or peak bone mass. And they also realized that it's very important to fight against osteoporosis. So, we have two types of osteoporosis. For men, we have senile osteoporosis. When the man is really old and testosterone is low, his bone mass is going to drop. For females, we have menopausal osteoporosis. So after menopause, females' bone mass drop. But it's not the same for every female, not the same for every male. If you exercise, you have the moderate exercise stress, then you're gonna retain your bone mass the more you exercise. So these marathon runners or these weightlifters who apply resistance to their bones, the bones, as we will learn now, they're gonna adapt by getting stronger. But if you don't, like they say, if you use it or lose it. If you don't use it, you're gonna lose it. Same thing with your brain. The more you study, the more you make it integrate information cognitively, the more it remains intact. Otherwise, dementia is gonna creep up into it. Okay? Okay. So we said stress. So not every single stress is bad. We need moderate stress is actually necessary. Physical stress is temperature. So we have stress is always hyper, hypo, and U. So hyper when there is excessive, and that is very bad. Too much salt is bad. Too little salt is bad. Too much sugar is bad. Too little sugar is also bad. We need the right appropriate amount. So hyper stress, that's a problem. And the opposite of hyper is hypo stress. And what we need is in the middle. We need homeostasis. That is known as the U range. I always draw this as the green. The traffic light, green is go. So that is where we are supposed to be. So green is where we belong. Green is where we belong. And then we have protective phase, yellow. That's adaptation. The yellow is the adaptation. So your body is going to try to adapt to the stress. If it succeed, we go down to green. If we fail, then we go to red. Red is now the disease. So if you go to red, that is where things are really bad. That is when we need, that is when death can occur. Red. So red, this line here, when you cross this red line, that's where the problems happen. And we will see that, you know, we need homeostasis is this green range. Homeostasis is this green healthy range. Homeostasis is the green healthy range. Okay, so what happens is we have different variables. One variable is, for example, blood sugar, known as glycemia. Glycemia. If you have too much sugar, that's a hyperstress, known as hyperglycemia. If you have too little, that is hypoglycemia. Well, both of them are bad, even though this is bad because it's hyper, plus, 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 and this is bad because it's drop, minus, 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 hypo. We don't want hyper. We don't want hypo. We want euglycemia. Another example is the temperature. The temperature must be 25. Temperature must be, 37 degrees Celsius. If it's too hot and it becomes 45 degrees Celsius, that's an example of hyper or hypo. Hyper what? Hyperthermia. When it was sugar, it was hyperglycemia. When it's temperature, is hyperthermia. What about if it's so cold, freezing outside? We drop from 30 to 20 or 15 degrees Celsius. Is that hyper or hypothermia? Hypothermia. Well, both are bad. The same way we don't want hypoglycemia, we don't want hyperglycemia, we want euglycemia. We also don't want hypothermia, we don't want hyperthermia. What do we want? Euthermia. Excellent. 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 You guys had good coffee today. Euthermia. Hold on. Now we're gonna learn how your body, when these fluctuations happen, it's gonna have to bring it down. So when it's too cold, your body has a system to restore things. When it's too hot, your body has a system to restore things. Let me give you one last variable. How about calcium? Calcium has a normal green homeostatic range, known as calcemia. If there is so much calcium in your blood, that is known as, excellent, hypercalcemia. If there is too low calcium in the blood, that leads to hypocalcemia. Well, both are bad. Too much calcium is bad. Too little calcium is bad. Why is too much calcium bad? Because calcium is like a key. Calcium can turn on enzymes from off to on. It can turn knife enzymes, degrade enzymes, proteolytic enzymes, metalloproteinase enzymes from off to on. There's a very dangerous disease known as acute pancreatitis. Acute pancreatitis, what happens is, the people alcohol in the body, it's going to convert into reactive oxidative species. That the liver is going to damage the body. That's why we have alcoholic liver disease. Calcium There are enzymes in the pancreas that are supposed to be off, but because there's so much calcium, it's gonna turn them from off to on. So now they're gonna start destroying everything nearby, and that is known as autocatalytic destruction of the pancreas by its own enzymes because there's too much calcium. They wouldn't never gonna be turned on, but because you have so much calcium, it turns them on. So that shows you how hypercalcemia is a problem. Hypocalcemia is also a problem. In hypocalcemia, what happens is you need the calcium for the action potential. We're going to learn more about that. You need the calcium for the electrophysiology of the heart. In order for the heart to pump and beat and contract, you're going to need the calcium. If you don't have enough calcium, you're not going to be able to mediate the electrical stimulation. You cannot pump the heart. You cannot pump the heart. You cannot pump blood. You cannot pump blood. The tissue is not going to have plus oxygen. It's not going to have plus nutrients for ATP. It's not going to have removal of all the metabolic waste and the high partial pressure of carbon dioxide. And then you're going to have death. So that's how you're going to go from something as little as a little extra calcium, leading to pancreatitis, destruction of the pancreas. A little bit depletion of calcium, and then you're going to have heart attack. So now we don't need hypocalcemia. We don't need hyperglycemia or hypoglycemia. We need euglycemia. We don't need hyperthermia or hypothermia. We need euthermia. We don't need hypercalcemia or hypocalcemia. We need eucalcemia. Okay, we need. Well, it doesn't work by need. There's a system to replace that. So now we're gonna go over these systems. Are we supposed to be out of here by 10? No, in half an hour. Okay. So in half an hour, when we move to that room, then you can have the break. Okay. You can have your break, and you go have the pancake, and you can have everything. But let me get this out of the way. Okay. So we need to know these three systems now, real quick. So how can we restore? How can we restore? How can we restore? My phone is working fine. You know, my phone, I tried to be smart. My battery wasn't working. So I bought battery from Amazon. I bought the repair kit. I said, I don't need somebody. I do it my own. I go on YouTube, how to install new battery. So I go, I open it. I take photo of myself doing this. I post it on my Instagram to tease all my friends. I'm genius. And then I'm trying to repair it. I did very good, up to 90%. The last part, I needed to put one clip, I broke it. So now, oh my God, I'm going to every single store. Kiss, they said, no, we only replace screen or battery. We don't fix things. I keep going here. I almost cried. And then I meet this guy. I said, oh, my lectures are on my phone. Please help me. He said, well, you had no business getting into something you had nothing to do with. Okay. He said, I don't come lecturing you students. You're not supposed to be opening your phone. That's my job. You're not supposed. So I said, okay, I'm sorry, can you fix my phone? He said, okay, come tomorrow. I come tomorrow. He said, come the day after. So he takes me around, and then it was repaired. But it's actually, everything was good. He just repaired that little clip. My phone would never last this long. Whenever I have it recording a lecture of mine, after 30 minutes, it would just die. But now it's going good. So if you take an adventure, it may go good. But there was a story back in the day of a crab. You know the crab from the sea. The crab said he doesn't want to live in the sea no more. He want to go live in the shore, and he want to go in the city. He's not. He want to go explore something. He went over there. Then they caught him and they ate him up. So that's what I did. That's when you go and get yourself into something you had no knowledge of. If you're a tech, you go to school for tech. But okay, I paid my price. So actually, I didn't save anything because when I added up how much it costed to replace the screen and how much I saved up, and then how much he charged me to repair it, I mean, it wasn't good. Okay, but I learned my lesson. Okay, it's not gonna be easy. Life doesn't teach you for free. But I mean, try, always try. That's my advice to you. You should try, but you should back up all your stuff first. before you try. You should back up in a flash all of your lectures, and then you should embark on your adventure. So now we have three systems to deal with. Let's begin with glycemia. Let's begin with glycemia. Let's begin with two patients. Patient A, who has, you guys want to begin with hyperglycemia or hypoglycemia? Hyper. Okay, so let's begin with one patient who comes in the emergency room with very excessive serum glucose. So that patient has hyperglycemia. So your body, to restore homeostasis, to restore you, green homeostasis. So what happens is your body has a sensor. The first thing that we deal with is a sensor. There's a sensor for every system. There is a sensor for excessive calcium, too little calcium. A sensor for